Let's bring you one. No, I don't know. 2, 3 more little short updates, little bite-sized updates. Yeah, because that's all we need for these. I think. So the first one is some computer scientists that are going into the structure of the SARS coronavirus spike, SARS coronavirus 2 spike rather. And we now know, we got this really exciting result at the beginning that, so all along for what we've known about spikes, we've known that the spike comes in a group of 3. So there's 3 of the same protein together, all bunched together. And we thought it was just like that. They were probably symmetrical, they were probably bunched up, and it's just a way they can hold on to each other, and you can have one thing that sticks out but gives you the ability to bind maybe 3 different receptors. Just sort of increases your chances, the way a multi-plug increases the number of plugs that you can stick into an outlet. But then we got the structure, and lo and behold, these 3 are not all the same. You have 2 of them that are locked down all the time. So 2 of the spikes are going to be in what we call the down conformation, and they're actually locked in that conformation together. And then one of them is going to be up like this. And it's not completely clear why one has to be up and the other's down, or if it has to be that way. But we do know there are a lot of antibodies that seem to block infection that are hitting the one that's up instead of the ones that are down. And so it looks like that may be at least an important way that the virus gets into cells. So who knows? And so what this paper finds is that there's one particular amino acid right down inside, and let's see if we can pull up the number from the picture here. There we go. Oh, those are nice pictures of it. And eventually, there we go. Okay, it's glutamine 564. So the spike is, I forget exactly how long. It's something like, let's see. 1,200 amino acids long? That might be right. I don't know. I could be a little off. But so spike has that many amino acids, and so you just number them 1 until the end. And everything in the first half is going to be called the S1 region, which is kind of the top of spike that's actually going to stick to the cell. So this is kind of the recognizer module of spike. And it might be able to stick to some sugar molecules. I think we talked about a heparin sulfate binding activity the other day. And we know it definitely also binds to the receptor ACE2. And but buried down in there, it's got these other 2 that are locked down. And it looks like the virus may actually need to have those locked down. And so the evidence from there is the absence of mutation at this site. So we've got lots of changes. The virus is able to change a lot of things about the spike, but there are some parts that don't seem to change much. So the first half of the spike is where most of the changes tend to be. And if you look at different spikes that are out there in the world, that's the part where they vary the most. They're under constant selective pressure. They're getting beat up by immune systems and everything. This is the part that's most visible. And so that changes fastest. And this amino acid, glutamine 564, is in that region as well. But so far, at least as far as I have seen in the papers that are out, there are not any reported changes at this site. So it looks like not only is the spike got 2 of these things locked down and 1 up, it looks like it may actually have to have 2 of them locked down in order to go about its business. So this is potentially another way that we could attack the spike and make it a little harder for the spike to do what it needs to do. If you could find a way with a small molecule to force 3 of these spikes to all be locked down, who knows? That might actually be a pretty good antiviral. Yeah. So there are, oh my gosh, this virus is like a super field day for chemists. They got so many things they could be making drugs against, and it's really wonderful. We're at the start of a long, well, I don't know, a large testing period. It doesn't have to be long. And there are a lot of good ideas out there, a lot of things that can work, and this is just maybe another one. So thanks very much, Dr. Ben Update.